is Joachim Siljedal. I'm one of the organizers here to the SDS project for this conference. First of all, before we have this session, I just want to mention that we have a Q&A with Kate Wong, Senior Editor of the Scientific American at 1 p.m. Calgary time. And this is a great opportunity, especially for PhD students and early academics. So please do not miss that. However, we have another great talk right now with Dr. Roberts, and I will leave the floor to you.
Great. Well, first of all, let me say a huge thank you to the organizers for putting together this conference. It's an amazing feat of work to bring everyone together and to see such fascinating and varied talks. So I'm going to talk a bit about how we use stable isotope analysis of fossil tooth enamel to reconstruct things about past hominin ecology and diet as well. And I think the best place to start then really is at the sort of ground floor, if you like, at the beginning. And this is the kind of fundamental principle of why do isotopes work? Why does stable isotope analysis provide interesting insights? And this is because sort of atoms appear in different forms. So atoms of different elements, whether that's carbon, oxygen, nitrogen, they have different forms, and these are called isotopes. And the differences between these isotopes, as you can sort of see here, is the different numbers of neutrons they have. And what that means is that you can have sort of slightly heavier isotopes. So that's, for example, carbon-13 versus slightly lighter isotopes like carbon-12. And in this very crude graphic, I'm going to sort of show you what that might mean for kind of reactions. And in a very simplistic way, carbon-12, for example, the lighter isotope tends to react at a kind of faster speed than perhaps carbon-13 that might be a bit more sluggish. And that's a gross oversimplification. But what this means is that you end up after a biological reaction, whether that's something like photosynthesis, whether it's an animal eating something and physiologically digesting it, there are different sort of ratios of these different isotopes at the end of each biological, chemical, physical reaction. And we measure this with this kind of delta symbol that many of you might have seen around. And it might look complex, but all it really refers to is the kind of relationship between the heavy and the light isotope in a sample that you measure, whether that's a plant, an animal, or a hominin, for example, versus a kind of known standard. And that's all that really means. And that's how we measure that in our mass spectrometer. And many of you, when you think of stable isotope analysis, might think of kind of bone collagen or this kind of protein, where we often do stable carbon and nitrogen isotopes. And this is done a lot in archaeology to look at questions of diet. But the problem is over the deep past is that, and also in more arid and challenging environments, collagen might not preserve very well. And as a result, that's why we look towards tooth enamel in these contexts. So tooth enamel is very solid. The bioapatite that makes it up is in a very strong prism structure that's very resistant to diagenesis. In this kind of beautiful diagram, you can kind of see the kind of brick wall nature of this. And importantly, we can look at enamel in a huge range of animals, ranging from hominins and then mammals, big and small as well. And importantly, these track kind of the relationship of diet or water sources or environments that animal is experiencing. So to take carbon isotopes first, you might have heard of this distinction between sort of C3 plants and C4 plants. Again, this might sound complicated, but all this actually refers to is the number of carbons that are in the first sugar that they make during photosynthesis. And so when we think of popular crops, things like wheat and rice are C3, and things like maize and millet are C4 crops. And these have very different carbon isotope values because the photosynthesis pathway works in very different ways and leads to different relationships between those heavier and light isotopes I mentioned before. Importantly, in kind of pre-agricultural settings, C3 and C4 plants are actually divided along environmental or climatic gradients as well, including within East Africa, which is an area where we're obviously particularly interested in this conference. And C3 plants, for example, in the tropics, such as in Eastern Africa, will be limited or they will dominate in tropical forest environments or woodland environments. And really C4 plants will only come to dominate in more open grassland or savanna settings, which means that we can use these distinctions to look at changing environments and changing diets in the past. And importantly, these distinctions in plants are passed up into their consumers. So in this plot, it's kind of showing data from Torre Serling, but you can see how these plants have very different non-overlapping carbon isotopic values, with C3 plants towards the left, the lower end of the spectrum, and C4 plants towards the higher end. Importantly, also, because C3 plants are much older than C4 plants, they have a lot of variation within them as well. So at the lowest end of the spectrum, we have plants growing under dense canopies, and at the higher end, sort of C3 grasses growing in more open settings as well. And this has been used for a long time in paleoanthropology and archaeology to look at questions of diets, what were hominins eating in the past? And you might be familiar with this kind of savanna hypothesis. It's been used a lot to say that through time, hominin diets shifted from a more C3 kind of pattern towards a more C4 pattern. And this was linked to kind of leaving the forest and heading out onto the savanna. Now, as you can see from this plot, which is sort of redone from Sponheimer et al. In 2013, it's actually much more complex than that. There is a broad shift from Ardipithecus ramidus through to Paranthropus boisei, but within that, there's a huge amount of variability, including within our own genus, that suggests actually the environmental usage of different hominins is far more complex and might actually vary quite significantly between different populations. In terms of oxygen isotope values, which is the other kind of measurement we can make on bioappetite, this reflects more the water that's being imbibed by animals as they eat or drink it. Here, the key factor is that kind of the lighter isotope during evaporation will more readily evaporate than the heavier isotope, which kind of makes sense, right? That the heavier isotope tends to stay behind a little more. And this means that kind of clouds that are forming above will have a sort of lower value than the ocean or the lake or whatever they're forming from. Then as they move inland and they rain, the rain is heavier because the heavier isotope tends to condense more than the lighter isotope. And so the rain is heavier than the cloud, as you can see here. And that will continue inland. And we can use that to sort of say something about not only the kind of precipitation that's happening, but potentially also things like local climate factors. And here, this is where we see this can be important because plants are obviously responding to, say, things like aridity and temperature in different ways. So we can see here, here's an open body of water that has a random value of 0, and here's a plant. And plants will have higher values because they're not only taking in the precipitation, but then evaporation is occurring from their leaves as a form of evapotranspiration. And importantly, as things get more arid, that gap between the groundwater and the plant will increase more because plants are so much more efficient at evaporation than, say, a water body. And so we can use the distinction between kind of groundwater sources and plants, as well as animals that tend to sort of get their water from these different sources, to say something about climatic aridity as well. So classic use of oxygen isotopes, they've been used to look at animals with different drinking strategies, different feeding strategies. So for example, hippos have been found to have very low oxygen isotope values, tend to be living in swamps, close to water, feeding on aquatic plants as well. Whereas giraffes are feeding high leaves on trees that are often more exposed to evapotranspiration. And so you can start to see how there are some oxygen differences in how animals get their water through time. So with these kind of basic principles in mind, I now just want to show you a couple of case studies that show you where these kind of different isotope systems can be used in hominin paleoecology and paleodiet, but also where some complexities emerge. So starting at the site of Pangaea Saidi on the coast of Eastern Africa, this has been in the news quite a lot recently. At this site, what's quite interesting due to its location is that it's very close to the coast. And yet there's also this very interesting sort of ecotone between tropical coastal forests and more open savanna grassland areas. And this kind of environmental context is important because at this site, there was found various Middle Stone Age and then later Stone Age technologies, as well as different forms of symbolic behavior. And elsewhere in Africa, these kind of behaviors have been linked to coastal settings, the idea being that there's lots of rich marine resources, or alternatively to savanna settings. And so at Pangaea Saidi, we really wanted to explore, there didn't seem to be much evidence of coastal subsistence. And so what was the broader prevailing environmental context that was sort of hosting populations that were experimenting with these different technologies and material culture? And so at a very basic level, what we did is we sampled lots of animal teeth through the different time periods. And we saw some different changes. So at the top of the carbon isotopes, this time the scale is flipped. So C3, kind of tropical forest, is on the bottom. C4, more open areas on the top. And you can see that there are broad changes through time. In MIS5, we see sort of more forest and wetter environments that shift towards kind of more drier open settings in MIS3 in the last glacial maximum before returning back to forest conditions. So this was quite interesting. What we can also see is that from MIS4 or MIS3, there's a big range. So there's definitely a range of an ecotone being used here through time. It's not just one environment or the other. But the problem with doing this, as has just been raised in a recent paper, is that obviously whatever animal I sample will in turn bias my isotopic value. So if I only sample grazers at a site, it's going to look like there's only grasses available, as well as if I only sample C3 browsers, I'm only going to find that element of the spectrum. As my old supervisor, Julia Lee-Thorpe, used to say, animals are not lawnmowers. They don't just sample the environment in a kind of random way. So we have to be careful when we're interpreting, and we have to also work with zooarchaeologists to make sure that our samples are relevant. And this was a recent paper brought out by Robinson. This was a plot he produced in a kind of more popular blog. And what he showed was that this link between C4 and C3 vegetation did not always hold for all animals. And this is because animals are being selective. They're doing different things. It works for some animals, but some animals, obviously like grazers, will simply not turn to trees or shrubs during any condition. And so returning to this plot then, this plot could be biased because it will be biased by whatever animals we've sampled. It also doesn't necessarily tell us whether humans are relying on forests or more open areas more or less. So to get around that, one thing we can do is, in this case, we separated browsers and grazers out in this isotopic system and then plotted them again. And what you can see here is that the actual changes are mainly happening among the browsers, right? This light green, the grazers, are not really changing in either carbon, maybe a little bit in oxygen as well through time. But the main environmental changes is what's happening to the forest and animals interacting with trees and shrubs. So that gives us a bit more detail when we start to look at this, and we can see the potential danger if we don't separate out animals with different feeding habits. Furthermore, if we want to look at what the actual humans are doing, it's important to actually sample the teeth of those hominins themselves. So for example, we had 2 loose Homo sapiens teeth. And when we actually run the isotopes from those, that we can see that although animals across a huge spectrum of diversity are present, the humans themselves are actually relying on towards the more forest woodland end of the spectrum. So you can see how it's important to not only sample the animals, but if we have questions particularly about what humans are relying on, then it's important to look at that direct information as well. Now moving to kind of where we can use oxygen isotopes. This is a case moving out of Africa, this time into Saudi Arabia. Tisselgada is a site dating to about 300,000 years ago. And here was found some of the earliest evidence for hominin behavior in the Arabian Peninsula in the form of cut marks and some stone tools. And one of the issues here is we're interested in looking at, well, how did hominins get into this area? Was it wetter in the past, this kind of green Arabia idea? And if so, how much wetter? And so we can use oxygen isotopes of animals to look at kind of what the environmental conditions were. But the problem is that oxygen isotopes can be affected by climate, but they can also be affected by water source. So they can, in this case of Arabia, you can have water coming from the Mediterranean, you can have it coming across Africa, and you can have it coming from the Indian Ocean. Each of these will have different oxygen values to start with. So if you see differences in oxygen values through time, it doesn't have to be climate linked. It could actually just be a shifting of the precipitation source, which is where that kind of element came in that I talked to earlier, where you have animals drinking from water bodies and animals getting their water from plants. And in this case, we can see things like obligate drinkers. So that's like equids, it's like ourselves. We have to drink from water sources to get enough water. But then there are animals like the oryx that actually get enough water from the plants. And we can look at these differences, the differences between these species, to look at actually how arid an environment is. And we can really narrow down those questions. So just an example here, you can see here in modern Arabia, here we have the non-obligate drinkers, so the oryx, and we have the obligate drinkers, things like horses. You can see that the difference is huge, and that's perhaps unsurprising because it's basically a desert environment. It's incredibly dry. These are animals living in kind of Eastern African savanna settings. You can see that the differences are much smaller. There's still a difference because it is still relatively arid during at least some times of the year, but it's much smaller than, say, modern Arabia. So this is how that system works. We can see that it's allowing us to get some insights into aridity. This is the site of Tisselgada. So we can see that 300,000 years ago, the environment or the aridity in this part of Arabia was much more similar to somewhere like Eastern Africa is today than it is to modern Arabia. So this allows us to get some insights into climate signals as well. We can also do sequential analyses on these different animals to look at if aridity is changing through time. So we can see that during times of the year, it got more arid before then becoming slightly wetter. This is kind of a classic, this curve here, one of these kind of classic seasonal curves where you're getting different conditions through the year. Interestingly, when we sample the elephants, we can see that they don't change that much through the year. And that's probably because they're really migrating significant distances, as elephants do in Africa today, which means that they don't really experience the seasonal areas of a given locale because they're constantly on the move looking for fresh vegetation. So turning to kind of older pie then and the work we've been doing here, we can use isotopes to look at kind of the feeding behaviors of animals and also potentially the climatic conditions as well. But we have to bear in mind these complexities that I've sort of raised along the way. So for example, this is an existing paper by Kevin Uno et al. And they showed that between these two intervals, which were linked to kind of older one technology or Acheulean technology, that really when they sampled these different animals, they saw very little difference actually in their carbon isotope values and oxygen isotope values through time. They argued that potentially there was a reduction in tree cover into kind of over the time period, but not that it was really significant. And again, importantly, they separated these out into groups so that they weren't just looking at this kind of big area, but at these different groups with different feeding behaviors. And again, you can see that actually when we look at this, we see even less evidence for kind of changes in climate or diet through time. And importantly here, what they did is they also added microwear and mesowear analyses, because this allows us to get, whereas isotopes giving a broad picture and will often be related to animal diets and animals won't be necessarily changing too much, the microwear and mesowear gives a more sort of short-term insight into diets. And through this, they were able to say that there was perhaps, again, a slight increase of a reliance on grasses over time, perhaps. This was the work we did recently in the paper led by Makeda et al. And here we did the enamel of the animals. Again, we were careful to try and compare our data from here to the data from the existing studies by looking at just the families that we had the same data for. And we could see that we sort of found a broad range that was very similar to what was seen in the Uno et al. Study, and a bit different from the Kanjira South data here that was a bit more further over. But importantly, what we were able to do is also do a very multidisciplinary approach. So not just using the isotopes of faunal tooth enamel, which gives you one insight into the animals that kind of ended up at the site, but also doing things like plant leaf waxes that I'm sure Bob Padalano will be talking about later, that gives a slightly different insight into the vegetation in a given catchment that is not just linked to the animals found on site as well. And putting this all together, we were able to see that actually over the time of occupation, there was a relatively variable change in environments through this time that was seen in the botanical evidence, in the plant wax evidence, as well as in this kind of relative variability seen in the isotopic evidence of the tooth enamel as well. And this is really, I think, the important thing with isotope analysis. It has a lot of strengths, but we also know its weaknesses. It's really perhaps not what would be considered state of the art right now, but I think there are positives to that because it's been applied since the 1980s. We know its problems. We know when it's worth doing an isotopic study and when it's not. It means that in terms of what we heard earlier, in terms of museum curation, we can start to make decisions as to when it makes sense to sample something or when it makes sense not to, because it's not going to provide useful information. And we can also create, or we can make sure that we have sensible questions that we can answer with this technique in the way that perhaps some of the latest methodologies are still building up their kind of, what I might call a kind of scientific maturity in this regard. But that's not to say that stable isotopes is static and not moving anywhere, because there was this recent incredibly exciting paper that showed that actually, although what I've been talking about is the bioappetite from the enamel, which we can look at carbon and oxygen isotopes of kind of the inorganic well-preserved area, it's also possible to actually extract the protein from the enamel itself. There's a very small amount of protein, as you can see in this diagram. And what's important is that if we can extract this protein, we can actually measure the nitrogen isotopes of it. And nitrogen isotopes are important because they've been used in things like bone collagen to look at trophic level, to see how much meat an animal is eating through time. And this great paper showed that in a feeding study, they were able to extract the protein of the enamel. And not only that, but the nitrogen isotopes of that enamel were reflecting the trophic level and how much meat an animal or an omnivore or a carnivore was eating in the past, which obviously holds incredible promise for paleoanthropology and archaeology moving forward as well, giving a whole new dimension of what we can learn about this very resistant kind of material using isotopic analysis moving forward. So that's it from me. Thank you very much for listening, and I'm happy to answer any questions.
Thank you so much for that amazing talk. We do have a few questions. So let me get those up. So the first one Are there other parts of the body except for tooth enamel that stable isotope analysis can be used effectively?
Yes, exactly. That's a very good question. It's often used on bone collagen. This is often seen as the kind of material of choice, if you like, because you can apply this carbon and nitrogen isotope analysis to that. There are two elements there. One is that the bone, it depends on which bone you sample as to which period of life that will represent. So ribs might represent the last 10 years of life, whereas something like a long bone or a femur will represent a much longer period of life. The other thing is it will only tell us the protein portion. So actually, it can over bias, if we're interested in plant parts of the diet, it can over bias towards things that are high in protein, like meat, like fish, and things like that. And the final problem is that often it doesn't preserve. I think its limit is about 200,000 years in Europe, for example, in a relatively temperate environment, but there are some studies that take it a little bit further. And so where we have arid conditions in the tropics, in some places, it's not even preserved back 5,000 years. So that's the other problem. And tooth enamel is a really nice alternative. And like I say, if we can get nitrogen isotopes out of tooth enamel, that's really a game changer in that regard, because that's really where tooth enamel has been slightly weaker. It's good at vegetation. It's quite good at climate conditions. It's not good at trophic level. But with this new data, maybe it will be in the future.
All right. Next question here. You said you can distinguish C3 and C4 plants because of their varying carbon isotope values. Can you distinguish CAM plants as well? And if so, what is the value of that?
Yeah, so this is a really good question, and one, like I say, with isotope analysis, we know our issues as well as our kind of benefits, and CAM plants are a big issue. They obviously, they can overlap, they can have C3 values, they can have C4 values, they can be in the middle. So they do cause potential problems where they might be significant. Often in the diets of humans, they're not so much of an issue. So if you're doing an isotopic study on humans, you can sort of, again, it comes down to really question. I think this is where it's quite important to know your environmental context, to know your ecological context, and to know your cultural context. Is it likely that CAM plants are playing a role or not? And if they are, can we account for that? In some places, we know that the values of CAM plants do actually still vary with environment, but it obviously is then a problem. So that is something we constantly have to bear in mind, that there are these issues that we have to try and overcome, or create questions or look at culturally meaningful questions, ecologically meaningful questions, where this isn't, where that might be an issue, but not enough to sort of stop us answering that question.
All right. We have two more questions. So second to last here. How does using carbon isotopes compare to other materials like strontium isotopes for stable isotope analysis?
Yeah, so I mean, carbon isotopes is primarily often used for diet, or in the case, because as I showed, there can also be these environmental gradients of C3, C4 plants that in turn can indirectly inform environmental situations. Strontium isotope analysis tends to be more used for mobility, to look at, it varies with geological ground rock, or as expressed then in soils and plants and things like that. So it's often used to look at, is an animal or a hominin moving around the landscape. In terms of basic analysis, also carbon isotopes, we need less material. It's at a point where even using a drill, it can be relatively unrecognizable. But the thing is that even with carbon and strontium now, with laser ablation, that basically puts in these minuscule pits using a laser. So actually now both, if you had that system, or you could use that system, for example, at the University of Cape Town, then it doesn't really matter. Actually, they both require sort of the same level. So yes, they're different systems, very much so, and often used to look into different questions. And in terms of, I guess, sample size, and that is also changing. So strontium is more expensive, is another. It's often right. A minimum of 50 euros per sample, but usually higher, whereas carbon is something more on the line of 5. So.
Understood. Yeah, that definitely would make a difference.
Yes.
All right, and then we have the last question here, quite fitting, I think. Where is the field headed? Are there new techniques that you can see revolutionizing stable isotope analysis?
Yeah, so I think the great thing about stable isotope analysis is that it's sort of become really, many archaeological projects now, archaeologists, I think, are taking on. It's been around for so long, and people are aware of the issues and not issues, even as non-specialists, that it's being factored into multiple projects, and it's really used as part of an archaeologist toolkit instead of perhaps being a black box that it's kind of you send off your samples and you get data back. I think archaeologists are increasingly involved in that. I think that's really important. And I think students looking at this should be able to, it's important in all archaeological science, I think, but archaeology students should be looking at learning about these techniques so that they can design projects that include them and that they know the pros and cons, that they don't just have to go send off samples to a specialist and get the results, that they can actually design the question themselves, that they know the pros and cons. And I think in turn, given some of the sort of talks we've had about sort of equity and equitable planning and collaboration, that's particularly important because then the archaeologists themselves can design these questions. And I think what I really like about stable isotope analysis is that I think that it's actually does that quite a lot now. It's due to its kind of maturity and people knowing the issues, and that is quite well known in general. I think that's exciting. It might not be revolutionary in many ways, but I think that in terms of collaboration and in that context, it is very important. I also think, as I say, it's still, although it's quite old and has been applied a lot, it's not standing still, and we're constantly getting new information, even from modern ecological studies. And as we can see from that recent feeding study, it looks like the whole sort of script of what we thought we could do with it is about to change in a big way. And obviously there needs to be careful testing of that new principle of the protein extraction from tooth enamel before it's just applied across very valuable remains. That's very important that that happens. But I think that's very exciting, and we're on the cusp of something that's about to provide potentially major insights into hominin diets, different animal diets through time as well.
Very exciting indeed. Thank you so much, Dr. Roberts, for answering the questions, for a wonderful talk. And I hope to have you in the audience for later sessions.
My pleasure. Thanks for listening.